Space and Science News with James. Today, we'll be talking about an incredible breakthrough in science that could change everything we know about climate change and medicine. What if I told you that tiny microbes, often overlooked and dismissed, are now being engineered to tackle one of the biggest challenges of our time, excess carbon dioxide in our atmosphere? These microscopic heroes are not just surviving, they're thriving on CO2 and transforming it into valuable pharmaceuticals. But how exactly do they do it? And what implications does this have for our future? Stick around, because you won't believe the fascinating science behind this process and how it could revolutionize both our environment and healthcare. So, let's dive deep into this subject of upcycling CO2 with tiny microbes, because why not? The future of our planet might just depend on it. Today, we're looking into a groundbreaking scientific discovery that could revolutionize how we combat climate change. Imagine if the very air we breathe, specifically, the excess carbon dioxide causing global warming, could be transformed into valuable resources like life-saving medicines. Well, researchers have engineered microscopic heroes that can do just that. In a world grappling with rising greenhouse gas levels, particularly carbon dioxide, we need innovative solutions. While reducing emissions is crucial, we also must tackle the CO2 already in our atmosphere. Enter the fascinating world of microbes, tiny organisms with enormous potential. Some microbes can make us sick, while others are essential for our survival. But today, we're focusing on a specific microbe that scientists have modified to turn harmful CO2 into a useful compound called mevalonate, a building block for pharmaceuticals. Join us as we explore this cutting-edge research that's bridging the gap between environmental science and medicine, potentially changing the way we view carbon dioxide and its role in our world. Get ready for a journey into the microscopic realm where climate solutions are born. Our planet faces a critical challenge, the relentless rise of greenhouse gases, particularly carbon dioxide, driving global warming at an alarming rate. As emissions continue to increase, the urgency for effective solutions grows exponentially. The impacts are already visible, from melting ice caps to extreme weather events, and the clock is ticking. While efforts to reduce emissions are vital, we must also find ways to remove existing CO2 from the atmosphere. This is where the incredible potential of microbes comes into play. These tiny organisms, invisible to the naked eye, play diverse roles in our ecosystem. Some cause diseases, others help us digest food, and many are crucial for nutrient cycling in the environment. Now, researchers are harnessing the power of specific microbes to combat climate change. By genetically engineering these organisms, scientists aim to create living factories that can convert CO2 into valuable products. This innovative approach could help us address both the excess carbon in our atmosphere and the demand for sustainable materials. In this video, we'll explore how scientists are using advanced genetic engineering techniques to transform microbes into powerful tools for carbon capture and conversion, potentially revolutionizing our approach to climate change mitigation. So, how do these microbial factories work? Scientists have long known that certain microbes can be genetically modified to produce useful substances. For instance, engineered bacteria now create insulin for diabetics, revolutionizing treatment for millions. But what if we could reprogram microbes to consume CO2 and create valuable chemicals? This is where Cypriavitis nicator H16 comes in. This remarkable bacterium thrives on a simple diet of CO2 and hydrogen gas, making it an ideal candidate for carbon capture and conversion. Its ability to grow using these basic components makes it a potential powerhouse for sustainable chemical production. However, there's a catch. While C. nicator can be genetically modified to produce new molecules, it struggles to retain these modifications over time. The genetic instructions, called plasmids, are often unstable in this species. This means that even if scientists successfully program the bacteria to produce a desired compound, the microbes might forget their new instructions after several generations. This instability poses a significant challenge for using C. nicator in large-scale, industrial applications. Scientists needed to find a way to make these genetic modifications more permanent and reliable. In the next section, we'll discuss the breakthrough that could change everything for C. nicator and its ability to produce useful products from CO2. Katalin Kovacs and her team set out to solve the plasmid stability problem in C. nicator. They focused on an enzyme called rubus-CO, which is crucial for the bacterium to utilize CO2. Their ingenious solution: They linked the new genetic instructions to Rubus CO production. This meant that if a bacterium forgot its new programming, it would also lose the ability to make Rubus CO, and consequently, its ability to survive. Only the bacteria that retained the genetic modifications would thrive and reproduce, passing along the stable plasmid to future generations. The results were impressive. These newly engineered microbes significantly outperformed control strains in producing mevalonate, a valuable 6-carbon molecule. By tying the bacteria's survival to the retention of the new genetic instructions, the researchers effectively created a self-selecting system that ensures the desired traits are maintained over time. This breakthrough represents a significant advancement in microbial engineering. It demonstrates a method to enhance the stability of genetic modifications in bacteria, potentially opening the door to more reliable and efficient microbial factories for various applications. In the next segment, we'll explore why mevalonate is such an important compound and how this research could impact multiple industries, from pharmaceuticals to sustainable chemical production. Mevalonate might not be a household name, but it's a crucial building block in both living organisms and pharmaceutical manufacturing. It plays a vital role in producing cholesterol and various steroid molecules used in medicine, including anti-inflammatory drugs and hormones. In fact, mevalonate is so important that it's often referred to as a metabolic hub in biochemistry. What makes this research truly groundbreaking is the scale of production achieved. This study produced the largest amounts to date of mevalonate from CO2 or other single-carbon reactants using microbes. This isn't just a scientific curiosity, it's a potential game-changer for sustainable pharmaceutical production. By using atmospheric CO2 as a raw material, we could significantly reduce our reliance on petroleum-based chemical synthesis while simultaneously helping to mitigate climate change. This approach represents a step towards a more circular economy, where waste products, in this case, excess CO2, become valuable resources. Moreover, the production of mevalonate from CO2 could lead to more cost-effective and environmentally friendly ways to manufacture a wide range of products, from medicines to cosmetics. In the next section, we'll explore the broader implications of this research and how it could revolutionize multiple industries. The implications of this research extend far beyond a single type of bacteria or molecule. The researchers believe their approach could be adapted to other microbial strains, potentially opening up a whole new frontier in sustainable biomanufacturing. Imagine a future where harmful greenhouse gases are routinely captured and transformed into life-saving medicines, industrial chemicals, or even everyday products. This could create a virtuous cycle where addressing climate change also drives innovation in multiple sectors of the economy. Moreover, this technology could be particularly valuable in space exploration or closed-loop systems where resources are limited. Astronauts on long-term missions could potentially use similar processes to recycle their CO2 emissions into useful chemicals, ensuring sustainability in challenging environments. The potential applications are vast. From creating biodegradable plastics to producing biofuels, engineered microbes could play a crucial role in transitioning to a more sustainable economy. This research also opens up new possibilities in synthetic biology, potentially leading to the development of novel organisms designed for specific environmental or industrial tasks. While there's still work to be done to scale up this process and explore its full potential, this research represents a significant step towards a more sustainable and circular economy. As we face the dual challenges of climate change and resource scarcity, innovations like this remind us of the power of scientific ingenuity. By harnessing and enhancing nature's own processes, we're finding ways to turn problems into solutions. These tiny engineered microbes represent a convergence of multiple cutting-edge fields: synthetic biology, climate science, and pharmaceutical research. They exemplify how interdisciplinary approaches can lead to breakthroughs that address multiple global challenges simultaneously. As we continue to explore and develop these technologies, we're not just finding ways to clean up our atmosphere, we're potentially revolutionizing how we produce essential chemicals and medicines. It's a powerful reminder that sometimes, the biggest solutions come in the smallest packages. While there are still hurdles to overcome, including scaling up production and ensuring long-term stability, the potential benefits are enormous. This research could pave the way for a future where our fight against climate change goes hand in hand with advancements in medicine and sustainable industry. Thank you for joining us on this journey into the world of microbial engineering. If you found this video insightful, please like, share, and subscribe for more exciting content on science and innovation. Thanks for watching. Remember to like, subscribe, comment, share and click that notification bell so you never miss out. See you next week.